Okay, I'll do a countdown. We're going to be starting up in 3, 2, 1.
Good afternoon, everyone. This meeting of the Advisory Committee on Plants and Animals is called to order on June 9th, 2022 at 1:36 p.m. My name is Darcy Oishi. I am the acting manager of the Plant Pest Control Branch. Well, we're in the introduction section of the agenda. I am the alternate to chair this committee if Dr. Helmut Rogg, the plant industry administrator, is unable to chair, which he is not. With me today in the room is Ms. Stephanie Easley with the coordinating group on alien pest species. I wanted to first start by letting you know of a few new individuals to the committee. We'll start with Dr. Samuel Gahn of the Nature Conservancy. I have a copy of the memo from Chairperson Shimabukuro Geyser appointing Dr. Gahn to the committee. This letter will be filed in the minutes. Dr. Gahn, please introduce yourself to the committee and to our audience.
To unmute. There we go. So aloha everyone, and I'm very pleased to be part of this committee. As you may or may not know, I served for many years on the Board of Land and Natural Resources, and so I'm familiar with many of the conservation issues. It will be very interesting to look at this through the lens of the Department of Agriculture. By way of background, you can look me up. I'm easy to Google, so I'll just leave it at that.
Thank you, Sam. We also have a new ex officio designee as alternate, Ms. Cynthia King from Chairperson Case with Department of Land and Natural Resources. I also have a copy of the memo designating Ms. King to the committee, which will be filed in the minutes. Ms. King, could you please introduce yourself?
Hi, yes, I'm Cynthia King. I'm the entomologist for Department of Land and Natural Resources, Division of Forestry and Wildlife, and I manage the Hawaii Invertebrate Program, which primarily focuses on stabilization and recovery of threatened and endangered and otherwise rare native invertebrate species.
Thank you. And we have another new member. Fish and Wildlife Service has appointed Mr. Joshua Fisher as an alternate for Ryan Pea, and Chair Shimabukuro Geyser has approved this request. I have a copy of the memo which will be filed in the minutes. Mr. Fisher, please introduce yourself.
Good afternoon. I'm Josh Fisher. I'm an invasive species biologist. I've been working in conservation in Hawaii for the last 18 years. 14 of that have been with the Fish and Wildlife Service, specifically focused on recovery issues associated with invasive species.
Okay, and let's round out introductions with our ex officio introductions. Department of Health. Grace.
Good afternoon. This is Grace Simmons. I'm the program manager for the Department of Health Vector Control Branch, and we basically are interested in the public health aspect of our invasive species and vector control.
Mr. Eisen from the organization formerly known as Office of Environmental Quality Control.
Yes, now it is the Environmental Review Program within the Office of Planning and Sustainable Development. I'm a planner in the Environmental Review Program, but actually pinch hitting for the director of OPSD, Mary Alice Evans, who's ex officio on this committee.
And finally, Dr. Hawes.
Aloha, I'm Maria Hawes. I'm a faculty member at the University of Hawaii at Hilo.
Thank you all for joining us on this Aloha Thursday. We're now on agenda item 3, comments from the general public on agenda items. We have 2 submittals for today that have elicited a significant amount of testimony. So I'll be limiting oral testimony to 2 minutes to allow the public to have feedback while still allowing sufficient time for the committee to discuss and make recommendations to the Board of Agriculture. Kevin, will you inform the public on how to provide testimony in this hybrid meeting? Again, testimony will be limited in time to 2 minutes. Kevin?
Yes, so to those who are attending this Zoom meeting for the Plant and Animal Advisory Committee meeting, if you're attending the Zoom meeting through the Zoom app or the Zoom or through a web browser, you know, if you want to give your testimony, feel free to click on the raise hand button next to your name. To those who are calling in to the Zoom meeting for phone, you know, phone audio, go ahead and type in *9 if you want to raise your hand and *6 to mute or unmute yourselves.
Okay, thank you. So as we have I believe we have a mix of both physical and virtual participants who wish to provide testimony. I'll be switching back and forth, but I'm going to start with written testimony. This is the old version without the numbers. Let's see. My apologies, I have all the stats written down. We have so we have received written testimony for both mosquito proposals being reviewed by the committee today and the board order. Because it is difficult to separate the two based upon how we collect written testimony, I'll just lump everything together. We have 95 in support. This is primarily for bird protection / prevention of the extinction of native bird populations through disease mitigation. We have some support due to vectoring of diseases impacting public health. And we have a few pieces of testimony supporting the cultural significance of birds. I will note that 2 of the letters of support are from State Senator Inouye and Representative Tarnas. We have 103 pieces of testimony against, with the bulk of those, about 94, focusing on genetically modified organisms or genetically engineered organisms. 9 are just generally against. There are a few pieces of testimony citing all introductions of anything are bad, with examples like mongoose, and a few that state there are no diseases of concern, and that we should eradicate cats instead, or that there is a lack of long-term studies. We also have some late testimony that was received after 4:30 p.m. yesterday. 7 of those are letters in support and 4 are against. I'll be now hearing testimony for the request to be reviewed by the Plants and Animals Committee, agenda item 4.1. Request to preliminary review the currently unlisted southern house mosquito, Culex conchifaciides, Diptera culicidae, for future placement on the list of restricted animals Part A by board order for immediate field release to suppress wild populations of Culex conchifaciides by the State of Hawaii Department of Land and Natural Resources. Please remember when presenting for oral testimony to state your name and any organizational affiliation clearly. Kevin, can you let in the first virtual participant, please?
So to the committee, I have Chris Farmer. Chris, could you please state your name and affiliation for the record?
Sure. Hello, committee. Thank you for this time. I am Chris Farmer. I'm the Hawaii Program Director for the American Bird Conservancy. And I want to speak strongly on behalf of the support for the recommendation to list these species. Hawaii is experiencing a conservation crisis, and the Wolbachia method that the Department of Land and Natural Resources and Department of Health have proposed is the best, safest solution to save these birds. All 3 of these mosquitoes were found widely in the state. The bacteria is here and widespread as well. And I'm really concerned that hearing about the GM opposition, because this is not a GM technique. There's no genetic modification. There is no manipulation of any of the genome. It is not anything like what you've heard about in Florida with a group called Oxitec. And this is reversible, so we have the ability to go very safely and slowly if needed. It's widely used and safely used for human health around the globe and on the continent. And so we, the Hawaiian group, I work with a group called Birds Not Mosquitoes, as well as with ABC, are building on millions of dollars of research and deployment, implementation, and extensive monitoring. And so this is the best option to save the Hawaiian honey creepers and our native birds. It's not a pesticide, it's not a toxin, nothing's killed, and there will be still additional regulatory oversight. There is going to be the Board of Ag's going to have an import permit. We're going to have additional permit conditions. This is just the next stage in an important regulatory process. But the birds are declining. We're going to lose species here very shortly if we don't start acting on these important management options. So I urge you to approve the recommendation from DOH and DLNR. Thank you very much. I know you're so busy, there's a lot of testimony, but if you have questions, I'm also available to respond to any other further questions.
Thank you, Mr. Farmer. Do we have anyone else wishing to provide testimony virtually? Christy Martin, please.
Christy, could you please state your name and affiliation for the record?
Yes, mahalo. Christy Martin with the Coordinating Group on Alien Pest Species, CGAPS. Mahalo for this time and this opportunity. CGAPS strongly supports these both proposals. As Chris Farmer said, you know, these birds are running out of time. This is the only available technique that we have to try to save some of these birds. Widespread birds like the non-native zebra dove are the ones that are really infected with these diseases, avian malaria and avian pox. When you see them, you can see that they look sort of sick. When the mosquitoes go to bite them, they can transfer them to the native birds. And as soon as it gets warm enough up on the top of the mountain where these mosquitoes can reproduce, those two things are going to come together, the mosquitoes, the diseases, and they're going to be spreading them to the native birds. We know this is happening. Our climate has been changing for a while, and we've seen these birds declining. So I urge you to recommend that these submittals be approved, and we just thank you for your time and your consideration. Mahalo.
Thank you, Christy. Let's take next submitter from the physical meeting, Stephen.
Give me just one moment.
Yeah, aloha members of the committee. This is Chelsea Arnott with the Hawaii Invasive Species Council as the program support staff over here. And we submitted testimony in support of both submissions, agenda item number 1 and agenda item 2. And just piggybacking on the previous testifiers on the importance of having these large scale tools developed and deployed to help prevent the spread of these invasive species, that the impacts are just going to be exacerbated by climate change. What we're talking about here is the extinction of native forest birds. And like Chris Farmer said earlier today, this is a tool and technique that's being deployed internationally and nationally to control and reduce large populations of mosquitoes, not only for human health, but for natural resources. And this is really our best and safest tool for reducing mosquitoes and saving forest birds from extinction and protecting human health along the way. So really appreciate the opportunity to testify on these two submissions. Mahalo.
Thank you. Kevin, do we have anyone else offering virtual testimony?
Yes, I have one person.
Ms. Woodside.
What's Woodside? So please state your name and affiliation for the record.
Aloha, this is Ulalia Woodside, and I'm the Executive Director of the Nature Conservancy's Hawaii and Palmyra program. The Nature Conservancy strongly supports the Department of Land and Natural Resources and Department of Health applications regarding the mosquito species that were mentioned. These permits will allow Hawaii to take steps towards developing and implementing a mosquito control program that uses Wolbachia bacteria to reduce mosquito populations throughout the state, which would have positive impacts on human health and the precarious state of our native forest birds. This work with Wolbachia is not genetically modified organisms or any type of genetic engineering. Hawaii's native forest birds highlight the extraordinary biodiversity of our islands. However, these species are facing extreme threats from avian pox and avian malaria being transmitted by introduced mosquitoes. Due to the increased temperatures as a result of climate change, mosquitoes are now expanding into higher elevations where birds had previously been safe from disease transmission. The expansion of mosquitoes is causing rapid declines in native forest bird populations. If we don't take action, we are going to see many more extinctions. We may be already too late for some. This is the way in which we will be able to save Hawaii's native forest birds. As each of these species that is endemic to Hawaii that's found nowhere else in the world goes extinct, a part of who we are and our identity as people of Hawaii also goes extinct. Once again, mahalo for the opportunity to support this vital effort to protect our native birds.
Thank you. Any other virtual testimony? Okay, seeing that this will be the last call for virtual testimony or any testimony for agenda item 4.1. Last chance for agenda item 4.1. If not, we'll transition to testimony for agenda item 4.2, which is a request to preliminarily review the currently unlisted yellow fever mosquito Aedes aegypti, Asian tiger mosquito Aedes albopictus, and the southern house mosquito Culex quinquefasciitis, Diptera culicidae for future placement on the list of restricted animals Part A by board order for immediate field release to suppress wild populations of Aedes aegypti, Aedes albopictus, and Culex quinquefasciitis by the State of Hawaii Department of Health. For those testifying, please remember to state your name and affiliation, and that we are limiting testimony to 2 minutes. Thank you. Do we have anyone who would like to offer testimony on the virtual side of the equation? I do note that Ms. Arnott's testimony was for both proposals. Okay, hearing none. Last call. Okay, then we'll close out testimony for agenda item 4-2 now.
I have one person. I have Christy Martin.
Oh, Christy Martin, please.
Christy, please state your name and affiliation.
And you're muted, Christy.
Aloha, thank you so much. Christy Martin with the Coordinating Group on Alien Pest Species. I just wanted to note that our testimony also did cover both agenda items. Mahalo.
Well, thank you for clarifying.
And I have Miss Woodside again, who would like to give her testimony.
Ms. Woodside.
Miss Woodside, please state your name and affiliation.
Aloha, this is Ulalia Woodside, the Executive Director of the Nature Conservancy's Hawaii and Palmyra program. Our testimony is in support of this as well.
Thank you.
And I have
Chris
Farmer.
Sorry, Chris Farmer with American Bird Conservancy again. Just want to clarify that we also are supporting both agenda items. I wasn't clear we needed to separate, but both the DOH and DLNR proposals are strongly supported.
Thank you, Mr. Farmer. Any additional testimony, please? Okay, I will be closing testimony for both agenda items, and with that, agenda item 4, review the requests for the committee. Our first order of business is an entomology submission with a request for the Department of Land and Natural Resources. Mr. Kishimoto, could you please enter the room and review the submission? Thank you.
Chris, please state your name and affiliation.
Good afternoon, Chairperson Oishi and members of the Plant and Animals Advisory Committee. My name is Chris Kishimoto. I'm an entomologist with the Hawaii Department of Agriculture's Plant Quarantine Branch. So I just wanted to say that I think for DLNR's request, I'll probably go over a little bit more in depth, just in case people weren't able to get their hands on the actual requests themselves. They're very similar. I'll go in depth maybe with DLNR's request since it's first, and then we'll do it a little bit more abbreviated for Department of Health's. But this is a request to, number one, preliminarily review the currently unlisted southern house mosquito, Culex quinquefasciatus, for future placement on the list of restricted animals Part A by board order for immediate field release to suppress wild populations of Culex quinquefasciatus by the State of Hawaii Department of Land and Natural Resources. 2, provided that Culex is placed on the list of restricted animals Part A, allow the importation of lab-reared strains of Culex that has been inoculated with strains of Wolbachia bacteria by permit for immediate field release to suppress wild populations of Culex quinquefasciatus by DLNR. And 3, provided that Culex is placed on the list of restricted animals Part A, to establish permit conditions for the importation and immediate field release of lab-reared strains of Culex that has been inoculated with strains of Wolbachia bacteria in order to suppress wild populations of Culex by DLNR. The applicant and importer in this case is going to be Suzanne Case, the chairperson of the Hawaii Department of Land and Natural Resources. The shippers are going to be Mosquito Mate Inc. in Lexington, Kentucky, as well as Verily Life Sciences based out of South San Francisco, California. And the commodity will be various shipments of the southern house mosquito, Culex quinquefasciatus, which has been inoculated with strains of Wolbachia bacteria. This project, just to reiterate, will not use any genetically modified or engineered organisms, and only male mosquitoes are going to be imported. Male mosquitoes do not bite, and they don't need to feed on blood or anything. These mosquitoes will either contain the same bacterium, Wolbachia species, which is already in Culex quinquefasciatus mosquitoes in Hawaii, or they'll also be inoculated with an incompatible bacterium, incompatible Wolbachia bacterium that is not native to the wild mosquito's current internal bacterial fauna. The presence of this different strain of bacteria within the male mosquito's reproductive system will render the imported male mosquitoes unable to successfully mate with wild females found in Hawaii. This process is called cytoplasmic incompatibility. And cytoplasmic incompatibility has been used with much success in other parts of the world to reduce mosquito populations and thus reduce the potential of transmission of mosquito vector diseases. The applicants intend to import male sexually incompatible mosquitoes for direct release into the environment to reduce current populations of this pest mosquito species, which are vectors of pathogens to Hawaii's native animals, including pathogens such as avian malaria, and which can vector West Nile virus, lymphatic filariasis to humans, as well as heartworms to dogs. Culex quinquefasciatus is an invasive disease-spreading mosquito that is dispersed across the Hawaiian Islands since its accidental introduction in the 1800s. The species is present on Hawaii, Maui, Molokai, Lanai, Kaho'olawe, Oahu, Kauai, and the Northwest Hawaiian Islands. Culex can thrive at sea level to up to about 4,800 feet in elevation in Hawaii. The mosquito is able to transmit pathogens to native forest birds, and the spread of avian malaria in particular has contributed to the extinction of more than half of Hawaii's endemic honey creepers and continues to pose a risk to the remaining species. So far, efforts to suppress Culex quinquefasciatus through utilization of traditional vector control methods, including using pesticides, have proven to be inadequate at a landscape scale and are problematic for other non-target state and federally protected invertebrate species. Current efforts to control mosquito vector disease outbreaks are limited to reducing mosquito breeding site locations and localized applications of various larvicides and adulticides. As for the bacteria Wolbachia, it's a type of arthropod endosymbiont, and this does not occur in humans. This also won't occur in animals as well. Approximately 50% of insect species naturally have this bacteria. Wolbachia are passed also from the females to their offspring. Wolbachia cannot be spread by the released male mosquitoes because, as I said just now, they can only be passed down from mother to offspring. If a male mosquito with one type of Wolbachia mates with a female mosquito that has a different strain of Wolbachia, the resulting offspring can be inviable and not develop into mosquito larvae. Wolbachia male-based insect control programs have been highly successful for reducing local mosquito populations around the world. One of the more recent results for a trial was in Fresno, California, and that showed a decrease of biting Aedes aegypti female mosquitoes by 68%, 95%, and 84% during the peak mosquito seasons in 2017, 2018, and 2019, respectively. So Culex quinquefasciatus mosquito eggs originating from Hawaii or collected from Hawaii have already been provided to Mosquito Mate and Verily for development and testing of cytoplasmic incompatibility. These mosquitoes have been crossed with female mosquitoes carrying that different strain of Wolbachia. And once these mosquitoes have taken up the bacteria, the mosquitoes will then be back crossed with a separate population of mosquitoes that were also originating from Hawaii over at least 7 generations to ensure Hawaii's wild mosquito genetics are greater than 99% similarity with wild mosquito populations in Hawaii. Those mosquitoes are the ones that will be shipped and released in Hawaii. Within Culex quinquefasciatus, the strain of incompatible bacterium will be Wolbachia WLA, Wolbachia WLB, or Wolbachia WPIP4. These Wolbachia bacterium are not present within the corresponding species of Hawaii's established mosquito population. The presence of this bacterium will make these males sexually incompatible with the wild established female mosquitoes. Existing wild type bacteria strains that may be imported could also be WPIP5, which is already found on all the main Hawaiian islands. Based on the prior use of this technology in California, Florida, and Kentucky, there are no data to suggest releases of these male mosquitoes to have a negative impact on agriculture, the environment, or public health and safety. all mosquitoes will be back crossed, as I mentioned, for at least 7 generations to ensure that greater than 99% of the Hawaii genetics will be maintained within the mosquitoes that are shipped to Hawaii for release. This back crossing will also mitigate the risks of infections, microorganisms, and parasites to the mosquitoes via vertical transmission, thus lowering the risk of the mosquitoes accidentally introducing a new parasite or pathogen here. As the intended importation of these mosquitoes only includes the importation of male mosquitoes that do not bite or feed on blood, the unintended importation of an acquired pathogen is also eliminated. Verification of Hawaii biotypes of this Wolbachia strains will be conducted on initial shipments of male mosquitoes to verify requirements have been met in collaboration with the University of Hawaii and the Department of Health. Due to automation and machine learning technology being applied to the sex sorting process of these mosquitoes, Mosquito Mate and Verily have not previously identified a female in a single release container during the course of projects in Puerto Rico and Fresno. In another example, a published study estimated that there is a probability of less than 1 female being found in 200 million males. To date, PCR monitoring of mosquitoes that have been collected from release field sites have not identified any female mosquitoes from the company Verily. And at least once per year, Mosquito Mate and Verily will also conduct longevity and competitiveness studies comparing the mosquitoes proposed for releases and wild type males. Data from previous trials demonstrated that Verily mosquito males' longevity and competitiveness is at least equal to wild type males. In addition to Hawaii's import requirements, both shippers and the receivers, importers will obtain additional permits as required by federal, state, and county agencies. This request was also submitted to our advisory subcommittee on entomology for their review and recommendations. And this is their comments and recommendations are as follows. So for the question of approval or disapproval of future placement of the currently unlisted southern house mosquito, Culex quinquefasciatus, on the list of restricted animals Part A by board order for immediate field release to suppress wild populations of Culex quinquefasciatus by the State of Hawaii Department of Land and Natural Resources. 3 of the 6, so only 3 of our 6 subcommittee members responded. All 3 recommended approval unanimously. For question number 2, if Culex quinquefasciatus is placed on the list of restricted animals, Part A, the recommendation of approval or disapproval to allow the import of lab-reared strains of this mosquito, which has been inoculated with strains of Wolbachia, for immediate field release to suppress wild populations of Culex by DLNR. Again, all 3 responding subcommittee members recommended approval. For the final question of whether Culex, if it's placed on the list of restricted animals, Part A, the recommendation of approval or disapproval to establish permit conditions for the import and immediate field release of these lab-reared strains of the mosquitoes with strains of Wolbachia inoculated into them for immediate field release to suppress wild populations of Culex by DLNR. All 3 responding subcommittee members recommended approval. One of the subcommittee members, Ms. Janice Matsunaga, did have a few questions which the Department of Land and Natural Resources answered. We weren't able to get it to you guys in time for it to get into your submittal packets, but I have their answers right now. So subcommittee member Matsunaga wanted to know what were the EPA and HDOA label directions for releasing the mosquitoes. So DLNR's response was the sites to be treated are currently going to be state, federal, and private wildlife conservation areas that contain Culex quinquefasciatus mosquitoes throughout the state of Hawaii. The method of application is going to be point releases by hand or aerial releases. The rate of applications are going to be, basically, they're going to do some trapping and surveying ahead of time to determine what the estimated population of culex is in that area, and that would determine how many male mosquitoes that they'll release. What they're trying to do is getting a ratio of 10 imported mosquitoes released for every 1 wild type male. The maximum number of applications is going to be 156 applications per release site per year, based on a maximum of 3 releases per week. At every treatment location, releases may be up to a year, with the intention of having multiple releases per week. If strong suppression is achieved, releases may be reduced in frequency at a given location, with releases starting at a new location at the same cadence. Thus, the total number of application days will be up to 156 during the year. If the permit is extended, then a similar rate of releases will occur. Estimated total acreage is going to be up to 40,000 acres of state, federal, and private wildlife conservation areas in the state of Hawaii. And the amount of mosquitoes released, DLNR is thinking of up to 208 million males per year. This is up to 4 million. They want to release up to 4 million males per week. Ms. Matsunaga asked, will these Wolbachia inoculated mosquitoes be classified as a biopesticide product in Hawaii? DLNR's answer is yes. They will be classified as a biopesticide by both the EPA and HDOA. Another question was, what quality control testing will be done in the laboratory in Hawaii just prior to release of the imported mosquitoes into the environment? And how will the results be reported? DLNR's response is that quality control will be conducted at the rearing facility in California, and measurements of the number of dead males will be estimated to inform, to make sure they have the optimal shipping, shipping conditions prior to their release. The results of quality control testing will be reported to HDOA, U.S. Fish and Wildlife Services, and DLNR. Quality control of both the Wolbachia type and the efficacy should be done outside of Hawaii to reduce any possibility of female releases, and thus lowering the release's efficacy. Another question was, how will the results affect releases into the environment? DLNR's response was, if quality control is significantly reduced after shipping, then this will lead to a reduced efficacy of the releases, which will in turn lead to an increase in projected prevalence of the avian malaria vector Culex quinquefasciatus. As Culex quinquefasciatus is an invasive organism, reducing its prevalence will not cause undue harm, and being ineffectual result in no increased impact to the native forest birds. For the question of, will there be routine monitoring of traps for incompatible adult females or larvae as proposed by Verily Life Sciences? DLNR's response is yes. Post-release monitoring is essential to maintain the efficacy of the tool. Both female and male presence in traps will be monitored and assessed for Wolbachia type. For the question of whether the applicant is applying for 1 million mosquitoes and how many mosquitoes will be in each shipment, what is the duration of time between shipments? DLNR's response is that they are applying for more than 1 million mosquitoes, about 204 million, 208 million is more likely. The number in each shipment is estimated using that 10-to-1 ratio of males, imported males versus wild type males, and that will be determined by the area being treated. Shipments could be as often as bi-weekly, depending on wild type densities upon initial product application. For the question of what is the plan and procedure for releasing into the environment, DLNR's response is releases into the environment will occur via pedestrian releases, pedestrian-assisted aerial releases, and aerial releases. Each of the modes is being included in environmental assessments and releases will follow aforementioned draft label criteria as well as standard operating procedures relating to management actions and helicopter operations for Division of Forestry and Wildlife field staff. For the question of how will it be decided how many mosquitoes are released at each time at each site, again, DLNR's response is that's going to be determined during initial surveys, and they want to maintain that overflooding ratio of 10:1. How will each site release be determined? DLNR states that sites that are of conservation are going to be based on priority due to the presence of the remaining populations of rare forest birds that will be prioritized for release by state and federal agencies to maximize likelihood of species recovery. And for the last question of is there a potential release site list available? DLNR's response is as stated above. The HDOA EPA permit application is for state, federal, and private wildlife conservation areas that contain Culex quinquefasciatus mosquitoes throughout the state of Hawaii. Initial releases will be conducted on priority forest bird conservation sites on Maui, followed by Kauai, and then the Big Island of Hawaii. Proposed project areas are to be specifically identified in the environmental assessment documents. An initial public scoping for the Maui EA has been completed, and the information regarding the East Maui project areas can be found online. Project areas on Kauai and the Big Island have not yet been finalized. However, the statewide environmental assessment will be completed, which will cover environmental compliance for all state and private lands across Hawaii. That's all I got. That's all I have. Thank you.
Mr. Kishimoto. Committee, do I have any questions for either plant quarantine or the applicant?
I have a comment. Earlier in the testimony, there was mentioned that this was a tool that was used for control of mosquitoes in various locations internationally as well as in the United States. However, I think I would like to make the distinction that the DLNR proposal may very well be the very first proposal to use Wolbachia for a conservation of natural resources purpose rather than a human health purpose. And in that sense, it will be an exciting precedent for the use of this tool as a natural resource management tool.
Thank you. Chris, I wanted to give you a break before asking you questions, but in highlighting the response by DLNR to the questions of Ms. Matsunaga, you're bringing up about it made me think about transportation issues. And I note that permit condition 11 limits importation through the port of Honolulu. I was wondering if DLNR had any comments on what kind of issues that they anticipate. I would anticipate the transportation of these insects from production sites in the US mainland are going to be significantly longer. If everything has to go through the port of Honolulu and they're intended to go to being released in areas on Maui, how this will impact the efficacy of the program, if that has been examined, or anything. So it's a two-pronged question. Part one would be for DLNR, is this permit condition restrictive? And part two would be for plant quarantine. Is there a need to, given that the intent, and I'll ask a clarifying question about this later, that I glean from the permit conditions and the proposal, if the intent is only for males to be released and introduced, is there an overriding need to route everything through the port of Honolulu?
So
yeah. You want to go first, Cynthia?
Sorry, I can, but I didn't mean to speak over you. So
no.
happy to have you go first.
Okay. Usually most imports of live animals go through Honolulu anyway as the first port of entry. It's where we have the most control over our imports. But also you have to take into consideration that both the ports of Maui and Kauai are not considered full ports of entry, which means that they can't receive things like this for inspection, being the first port of entry.
But I note that a board order could change that.
I'm not sure if a board order could change that, but I think it has to definitely be a decision by the Board of Agriculture.
Okay, and how will all of these insects be moved? Is it going to be air freight? I'm assuming it's air freight, but is it going to be a priority parcel carrier like FedEx and UPS, or will it be a straight air cargo shipment? Just curious.
That's probably something Cynthia could answer better.
I actually can't answer the question about the actual carriers. That's something that I have not discussed, but there might be someone else in the room from that might be willing to, yeah, Josh Fisher, I think, might have a answer for you on that. But wait, but really quickly, just because Darcy did direct one other question to us, which is that we do know that having additional time where mosquitoes have to transit through the Port of Honolulu, undergo inspection, and then move on the neighbor islands for potential deployment or for actual deployment as planned, that does increase the time that they're in less ideal conditions, and it puts them at greater risk of higher mortality and lower survivorship or effectiveness in the field. So it's not to say that we can't do it. And I think my working assumption has been, or DLNR's, after having conversations with Department of Agriculture, was that that everything would be just going through the Port of Honolulu by default. But if there was a means by which that could be altered, then I would say DLNR would certainly be supportive of that.
What's the farthest distance Verily or Mosquito Mate has moved mosquitoes by air for releases? And I guess not just distance, it'll also be time from collection from their rearing facility to release in the field.
Well, the longest distance that I know that has currently been traveled is from, that I believe has, and so I can be corrected, is from California to Puerto Rico, where Verily has been undergoing a collaboration. And I think that the total estimated time that the mosquitoes could be in transit is about 24 hours, give or take. But less time in tiny chilled containers is typically better.
So adding on a movement from Honolulu to any other island would potentially push us beyond the 24-hour period, or no?
That remains to be determined. Sorry, I was just going to say that remains to be determined. So for the Puerto Rico applications, they did use FedEx, and that worked. As Cynthia alluded to, there is a time constraint that you want to do the shipments within, like 24 hours. I think there is maybe an upper limit to that. And so, various delivery services will have to be looked at, and how to best minimize the travel distance. Certainly mosquitoes coming from California here would not be as long versus shipments coming from the East Coast. So it's to be determined, but all the options would be on the table.
Jonathan, I'm assuming you're raising your hand to answer the question.
Yeah, so I'll be real quick. Jonathan Ho, Inspector and Compliance Section Chief for the Plant Quarantine Branch. So I guess a couple things. The the board could determine another port of entry, but that creates a logistical problem with regards to inspections, particularly from the fact that we don't have a lot of staff. So the mode of entry, so I guess the actual, mode of entry is going to play a specific role. For example, FedEx only comes through the port of Honolulu. You can't, there is no direct flight to an outer island. They don't do that. So that would be impossible. So the carrier is going to really depend. If you're doing air cargo, obviously that would be something different. However, you know, we deal with live animal shipments all the time. They're all required to come through Honolulu, and they're all done well within 24 hours, particularly now that COVID is over. So you get shipments that'll come in, you know, through Hawaiian Cargo. For Kauai, for example, they come in, they come for inspection. As soon as the inspection is completed, they're on a plane going to that island. So, I mean, you're talking maybe an hour or two, 3, depending on obviously the flight time, but within 24 hours is no ifs, ands, or buts guaranteed to occur, provided, you know, that there's not some crazy weird shipping issue. So I think just to kind of help, I think, with regards to that, because the port of entry issue is somewhat complicated.
Okay, thank you, Jonathan. Dr. Hawes, you've been patiently waiting.
That's fine. I just want to make a minor point that there's 2 misspellings in the first 2 clauses related to the subject, the request, where it looks like the second usage of the species name has been misspelled. I'm only saying that because in case anybody wants to refer to that language and if they make a motion.
Thank you for catching that. Mr. Kishimoto, I've got another, oh, I'm sorry, Cynthia, you got your hand raised.
Thanks. Yes, I just wanted to follow up really quickly and just note that that 24-hour time period doesn't just have to include getting a parcel physically to a neighbor island, but also then out into the field and then deployed. And so, and if that is being hiked out on foot in some areas, if that's via helicopter or other aerial deployment, that remains to be seen. That could be faster in some cases, but I just wanted to point out that there is potentially quite a bit of additional transit time just to get the mosquitoes to remote field sites where the last of our remaining forest birds take refuge.
Darcy, you're on mute.
Thank you, Cynthia, and thank you, Kevin, for pointing out that I was muting. I was talking just to myself. So, Mr. Kishimoto, I note in a few permit conditions, there's some potential conflicts. I'm assuming the desire is to make sure that only males are imported. So, given if only males are imported, perhaps we need to eliminate progeny from permit conditions 8 and 16.
That's a good catch, Darcy. We can do that.
Okay, thank you. And then on permit condition 6, it says maintenance by trained or certified personnel designated by the permittee. Is it the intent of plant quarantine branch to know who is certified and trained? Should we be modifying the permit condition language or expanding it to reflect reporting, or is the intent for that to be captured within the biosecurity plan requirement?
I think, at least for me, it can be either or. It would be nice to know who the trained staff is, and they can supply us with a list, but it doesn't necessarily have to be a specific list. It can be you know, inside their reports as well.
So I think it would be cleaner if we had a modification to the permit condition language so the applicant knows the expectation. Would that be a problem for the applicant? Thank you, Ms. King. I saw the head shake no. Any Okay, I also noted in the submittal that it said DLNR will be doing an EEA at a future date, and I heard, Chris, during your your presentation that a partial EEA was done for Maui. What's the timeline for completion of the EA? Cynthia?
The timeline for the EA to be released as a draft to the public, hopefully I'm not getting the terms wrong, is in September of this year for the East Maui area. There's and there's a parallel process going on for the Hawaii proposed project area. And then there will be a third EA to cover statewide. So there will be a total of 3 EAs completed for this.
And some of our testifiers have noted, and certainly the abundance of written testimony against this proposal are tied to concerns over GMO or genetic engineering. I'm assuming the EA will help address some of those concerns.
That's certainly the goal, to put it through the process and make it publicly available so that folks have an opportunity to learn about the project and express any concerns that they might have. And I just checked my notes really quickly. The draft EA is actually supposed to be published in August, not September.
Okay, thank you. And will there be other outreach or education opportunities to get the word out that this is not genetically modified organisms, or is there a plan on the part of DLNR to educate the general public about what this really is?
That's a great question. So the DLNR is part of a multi-agency partnership called Birds Not Mosquitoes. And so while DLNR doesn't specifically have outreach staff directed to this project at this time, we're a partner in this consortium, which has emphasized a great deal of outreach and education to the public, as well as legislators and community members. And that has been an emphasis for a long time. Obviously, there's still a lot of work to do out there based on some of the testimony that we saw today from concerned members of the public, and that's understandable. So it's a good reminder that that's an important task, and it will definitely be pursued. Recently, there was additional funding allocated towards this, towards implementing incompatible insect technique in Hawaii, and a portion of those funds will certainly be going to continuing outreach.
Okay, thank you. Do we have any other questions for the applicant or plant quarantine?
I don't have a question, but I do have a comment with regard to what Cynthia just said about DLNR and its public outreach. I know that a couple of days ago, Dan Dennison released a news release that indicated that there were no GMOs or GEs involved in this particular proposal. So I think it's part of the ongoing multi-pronged outreach and public awareness efforts that are going on in the Birds Not Mosquitoes group.
That's a good point, Sam. Thanks for the correction. And certainly we have done other, DLNR has issued other press releases at different stages as the interest in Wolbachia for forest bird conservation has evolved.
Yeah, I just didn't want you to not take credit for good work that has already been done.
Or give credit to Dan. Yes, we need to do that.
Darcy, we have Joshua who would like to address the committee.
Yeah, I think my hand's hidden by my logo.
Yeah, it was.
Yeah, and I just wanted to know, just reading through all the testimony that was received, I think one of the recommendations I'm gonna bring back to the communication groups, or those folks working on outreach, that they're gonna have to broaden to different audiences, because it seemed to me that there was a very large farming contingent or farming community that had submitted testimony, and so I think that outreach could be extended to those audiences. So that's going to be one of my recommendations to the outreach group.
Thank you, Joshua.
Yeah, if I can comment on that point, it really is interesting that the rise of anti-GMO sentiment in Hawaii was largely out of the farming and pesticide community, and so maybe it isn't that surprising that that would be the origins of this current anti-GMO and then conflation of mosquitoes with GMO.
Any other points of discussion? Do I have a motion?
I would like to move to approve that the DLNR item with the corrections and modifications that you and Dr. Haas mentioned.
Okay, so that would be modifications to permit condition.
And the correction of spelling.
Yeah. Permit condition 8 and 16. In terms of elimination of the word progeny. Permit condition 6 to specify the kinds of that the applicant is to provide a list of individuals that have received training, and the typographic errors in the title.
Yes, so moved.
So any second?
I'll second it.
Any further discussion? Being no oh, yes, Cynthia.
I was trying to scroll through, and I just wanted to clarify one point with Chris Kishimoto. I was trying to look in the original application, and I didn't see a reference, but perhaps I missed it, to having quality control upon arrival at University of Hawaii. I do believe that that was a condition from the previous permit, but I wasn't clear that I don't believe we had included that in the most recent application because of the quality control and then direct release approach. So I just want to verify if it's in the application. I don't have any concerns about it, but if it was a carryover, I just would like to see that potentially corrected.
As far as I know, that was in the request you guys submitted, so we kept it in there. If
I'm
you want,
glancing through it, I just don't see it.
I don't recall seeing anything about the university, Cynthia.
Yeah, I don't believe we put them on the hook for that, so to speak. I think they might be surprised to hear that we obligate them to that. So that's my only concern.
So you would like that condition added?
I would like that to not be included.
Okay. So it is fine as it is without it, correct?
Okay. Sounds good.
And then permit condition 7 also notes progeny, Mr. Kishimoto.
noted.
So we have a second from Dr. Hawes. Any further discussion? Okay, let me call for the vote. Chair votes aye. Mr. Eisen?
Aye.
Ms. King?
Aye.
Dr. Gahn?
Aye.
Mr. Fisher?
Aye.
Ms. Simmons?
Aye.
Dr. Hawes?
Aye.
Motion carries. Since we've been at it for a little over an hour, how about we take a 5-minute break and reconvene to address the second permit condition, the second proposal. So let's all be back 2:45 p.m. So I'm reading. Are we having fun?
Sorry, I was getting lost in too many open PDFs on my desktop. I try to organize them.
You're gonna see how I celebrate by throwing all the testimony up in the air and watching it fall.
You're supposed to throw it through the shredder first, and then throw it up.
Well, with 200 pieces of paper,
You might be have to shredder for a while.
I'd have to shred for a while. Okay, we'll have to wait for at least Jennifer to come back since she's legal counsel.
Oh, no, I'm here.
Oh, okay.
My video stopped.
Okay, great. We are all here. Chris, are you here? I won't start without you. Okay, then let's go back live.
Darcy, we're live.
This is the meeting of the Plants and Animals Advisory Committee meeting is back in session at 2:51. We're ready for our second agenda item 4-2. Mr. Kishimoto. Chris, you're muted.
Sorry, thank you. This submittal is going to be significantly quicker since it's very similar to DLNRs. But this is a request to, number one, preliminarily review the currently unlisted yellow fever mosquito Aedes aegypti, Asian tiger mosquito Aedes albopictus, and the southern house mosquito Culex quinquefasciatus for future placement on the list of restricted animals Part A by board order for immediate field release to suppress wild populations of Aedes aegypti, Aedes albopictus, and Culex quinquefasciatus by the state of Hawaii Department of Health. Number two, provided that the yellow fever mosquito, Asian tiger mosquito, and southern house mosquito are placed on the list of restricted animals Part A, allow for the importation of lab-reared strains of the mosquitoes inoculated with strains of Wolbachia bacteria by permit for immediate field release to suppress wild populations of those mosquitoes by the Department of Health. And three, provided that the yellow fever mosquito, Asian tiger mosquito, and southern house mosquito are placed on the list of restricted animals Part A to establish permit conditions for the importation and immediate field release of lab-reared strains of these mosquitoes, which have been inoculated with strains of Wolbachia bacteria by permit for immediate field release to suppress wild populations of these mosquitoes by the Department of Health. The importer is going to be Dr. Elizabeth Char, who is the director of the Hawaii Department of Health. The shippers again will be Mosquito Mate Inc. out of Lexington, Kentucky, and Verily Life Sciences out of South San Francisco, California. And the commodities will be yellow fever mosquitoes, Asian tiger mosquitoes, and southern house mosquitoes. As I said, this request is very, very similar. One of the only differences is that there are going to be 3 different mosquitoes that the applicants are asking to be brought in, and they're going to be used for, instead of trying to protect native forest birds, they're going to be used for human health purposes and trying to reduce those mosquito populations, especially if there's an outbreak of mosquito vector disease. Once again, this project will not be will not use genetically modified or engineered organisms, and only male mosquitoes are going to be imported and released for this project. And male mosquitoes they don't need to blood feed, and they don't bite anybody. These three types of mosquitoes are very important vectors of human pathogens like Zika virus, dengue virus, chikungunya virus, yellow fever virus, West Nile virus, and lymphatic filariasis, as well as vectoring heartworm for dogs. This request was submitted to the Advisory Subcommittee on Entomology for its review and recommendation. The Advisory Subcommittee recommendations and comments are as follows. I'll note that only for the subcommittee comments that you have, it shows only 2 subcommittee members responded. Since you guys have gotten this request, 2 more subcommittee members have commented, and I'll let you guys know what their comments and recommendations are. But for the recommendation of approval or disapproval for future placement of the currently unlisted yellow fever mosquito Aedes aegypti, the Asian tiger mosquito Aedes albopictus, and the southern house mosquito Culex quinquefasciatus on the list of restricted animals Part A by board order for immediate field release to suppress wild populations of Aedes aegypti, Aedes albopictus, and Culex quinquefasciatus by the Hawaii State Department of Health. As you can see in your submittals, both of the responding subcommittee members recommended approval. I have recommendations from Dr. Peter Follett and Dr. Dan Rubinoff. Both of those subcommittee members also recommended approval. For question number 2, provided that Aedes aegypti, Aedes albopictus, and Culex quinquefasciatus are placed on the list of restricted animals Part A, for the recommendation of approval or disapproval to allow the importation of lab-reared strains of those mosquitoes, which have been inoculated with strains of Wolbachia bacteria for immediate field release to suppress the wild populations of those mosquitoes by the Department of Health. All 4 responding subcommittee members recommended approval. And for the last question, provided that Aedes aegypti, Aedes albopictus, and Culex quinquefasciatus are placed on the list of restricted animals Part A, the recommendation of approval or disapproval to establish permit conditions for the import and immediate field release of those lab-reared strains of mosquitoes which have been inoculated with strains of Wolbachia bacteria for immediate field release to suppress wild populations of those mosquitoes by the Department of Health. Again, all 4 subcommittee members that responded recommended approval. Committee member Janice Matsunaga had a couple of questions, namely in question number 3. She wanted results of the quality control test done in the laboratory prior to release. She wanted a list of the intended release sites by locality and island. Again, the estimated number of individuals to be released, the number of releases that occur at each site, and plans on future releases. So basically the mosquitoes for the release under this request, as mentioned, undergo continuous quality control testing at the shipper's facility, as described in attachment 3, questions 1, 2, 3, and 15. And the post-release monitoring results for incompatible adult males and for females and larvae should be included in the report in permit condition number 18. That's all I have. Thank you.
Thank you, Mr. Kishimoto. Do we have any questions for plant quarantine and or the applicant? Aside from my questions. Okay. Go ahead, Mr. Eisen.
I was wondering, similar to what you asked in the first one about the status of the EA for this proposal. Is there going to be, what is the status, and are there going to be multiple, such as with the previous proposal?
So Tom, this is Grace. We are in the process of letting out a request for proposal to have someone conduct an environmental assessment. In terms of timing, you know, it just depends, but I would say sometime early next year, we could get a full report ready. It would encompass the entire state, not just Oahu or a specific island.
I would suggest looking and leaning heavily on the EA that DLNR is proposing that would presumably be dealing with some of the very same issues, make the job easier.
We've modeled our request for proposal based on what's already been released by DLNR. So we're learning a lot from their process. And so we've been working very closely with them on how to fashion the proposal.
Any other questions? So Chris, note, I have the same concerns over permit conditions for the Department of Health proposal as I do for the Department of Health. It behooves me to ask as it relates to permit condition 1. And I know this is not really likely, but I do want to ask the question, you know, permit condition 1 limits releases to islands that do not, that do have populations of these target mosquitoes, right? So what happens as we progress down the line and we are, if we are becoming successful in this very optimistic scenario for especially larger islands, but if we no longer are able to detect mosquitoes in our monitoring programs, are they going to be, is Department of Health going to be prevented from releasing, which may then impact the long-term success of the project? So we know eradication is the hardest step of the process. It takes more and more effort. So if, and this is a question both to health and to plant quarantine, how long of no mosquitoes are we supposed to being detected? Is it before eradication is declared, at which point you no longer can release, if I'm reading the permit condition correctly? And I didn't ask this of DLNR because with DLNR's focus on natural areas, you'd always have a reservoir in surrounding areas of culex that this situation wouldn't be coming up.
So this is for permit condition number 1, Darcy?
Yes.
And you're asking if you're asking about if it's only going to be used for the islands that have these mosquitoes already established?
Yeah, and so, you know, we've been flooding millions of mosquitoes onto, say, a small island like Lanai for 10 years. And surveys aren't showing overt signs of mosquitoes. Is Department of Health going to then be prevented from releasing these Wolbachia incompatible mosquitoes, which might then mess up the actual project, so the mosquitoes might come back because they're not at an overtly detectable level at this point in time based upon the survey methods being deployed?
So I think we included some of that language in response to DLNR and DOH that's some of the language that they included for their specific permit conditions. I think we've got to discuss with both DLNR and DOH if that's, you know, something that they've thought about and have considered. I would say that due to the fact that like only sterile males will be released, the risk is low to, you know, even if we have undetectable levels until we declare eradication, that it would be okay, I think, with the Department of Ag if they continue to release on those islands.
Jonathan?
Yeah, so Jonathan Ho, Inspection and Compliance Section Chief. Yeah, so I think just to add to that, you know, the condition does state, you know, again, a population, not necessarily establishment. So, and clearly the intent of this is for suppression and or eradication. And clearly, you may have a subdetectable level, and at that point, you know, the agencies that are doing that particular work, you know, would obviously make that call. And at that point, I think I won't speak for them specifically, but if there is no mosquitoes to control, they're probably not going to be releasing mosquitoes for something that they're not going to be controlling. So they would be basically stopping the project on their own. And if a detection is made, I think, you know, obviously surveying will occur. And if it's a one and done, obviously that determination of whether release is going to be needed or not will be made. And again, if you find, you know, 3 or whatever what you're determining is a population, at that point, it's there and you can release it. So I guess I don't look at this condition as being restrictive to the standpoint of their ability to release, because if it is eradicated and there are absolutely no mosquitoes, and I think that, you know, we would obviously hope that could occur. Should there be another detection, these conditions to me would be appropriate, because again, you're doing it for the same purpose. You find another population, you release mosquitoes to suppress it. So I guess I don't see it really being an issue. It's not necessarily whether they are established. It's, do you have a population? You know, what do you consider population? You find larvae, clearly they're breeding, treat. I mean, again, I'm not an ecologist or whatever, but I mean, I think that's clearly the intent of what's being done.
Thank you, Jonathan. Cynthia?
Thanks. I was just going to follow up. I just checked the original submission and I see that the Department of Health had included the statement that I added into the chat, which identifies if there is a known established or incipient wild mosquito population. And just to clarify for folks the differences here, Aedes aegypti is only found on the Big Island. And while populations of Culex quinquefasciatus and Aedes albopictus are really widespread and we basically have no hope of eradicating them, Aedes aegypti presents a target that could potentially be eradicated using incompatible insect technique. And it would be a potentially really great value considering that they transmit dengue fever, and we've known that Big Island is a place where dengue fever can really proliferate when there is disease that's introduced to that population. So I think it's important to make the distinction and not get rid of any criteria that says that we can't import these mosquitoes to islands unless there has been an established population, incipient population. I guess my question for Darcy or for Chris is it valuable or too limiting to put some sort of time frame on that? You know, if there hasn't, you know, within a period of X amount of years, that control efforts could continue, because we know for lots of different control programs in either agriculture or another invasive species context that that's important to have zero detection for sometimes several years before you can claim eradication. So this is really me asking a question though of what kind of language would you want to see as regulators though that would be defensible? Say if members of the public said, we don't want any more mosquito releases, they said they didn't find any last month, that would enable Department of Health to continue those operations to do that. that final bit of work that's so crucial. So your thoughts, I'd be really interested to hear.
I was thinking about, that permit condition and what you guys were talking about as well. And my question would be, how long would it take, what is the time frame for, number of, I guess, for the length of time you guys don't detect any mosquitoes to when you actually declare eradication, should that ever happen? And, until you guys declare eradication, I would think that it would be allowable and okay to keep releasing sterile males.
Darcy, you're on mute.
Josh, does your question or answer relate to the question posed right now?
Yeah, I think so. I wanted to just bring up the fact that, this tool is used for suppression of mosquito populations. It isn't intended for eradication. It could be done if the like as Cynthia said, the populations are small and finite. But I think also where this tool comes, efforts in the state have been trying to get more port security, biosecurity up and doing detections at our ports of entry, like airports, mosquito surveillance. And, a tool like this comes in handy. So for instance, if the state was to detect Aedes aegypti around Kahului Airport, there are wetlands that are around there with other wildlife. And so your options of doing a rapid response to address that incipient population are limited. And so a tool like this, which doesn't have really any non-target issues, would be ideal for a scenario like that. And so the verbiage of an incipient population, I think, is good. And especially when you have those very, very small occurrences of mosquitoes, it's very hard to detect them, to trap them and monitor them in the very beginning. And so I think having some kind of flexibility related to incipient, or as Chris had referred to before, like eradication is declared, I think is appropriate.
I see Christopher Jacobson of the Department of Health has his hand raised. Kevin, can you let Chris in? And Chris, please introduce yourself to the committee, please.
Good afternoon. My name is Chris Jacobson. I'm an entomologist on the Big Island for the Department of Health. I raised my hand as Cynthia and Josh began to speak, and they addressed a lot of, I guess, the rationale that the Department of Health has around incipient populations, most specifically Aedes aegypti. There have been introductions or detections on Maui as well as on Oahu, and potentially a tool like this could be used in those kinds of situations. At this point, the Department of Health certainly does not have ambitions to eradicate existing populations on the island. It would be a tool to be used mainly for increasing public health strategies and measures in response to arboviruses that are being transmitted on the island. So the area in the footprint would likely be very low and would be discontinued when public health threats are diminished.
So, Mr. Jacobson, how long, and I know this is more of a theoretical question based upon how much years of experience you actually have in releasing incompatible mosquitoes. How long would you think there would be a need for to release introduced Aedes aegypti carrying incompatible Wolbachia to suppress Aedes aegypti detections that may have happened on Oahu or Maui in the past? Would that have been weeks or months or years?
My guess would be months, just because when you're trying to eradicate, you want to have a degree of certainty and understanding the fact that detection of very low populations is really challenging for mosquitoes. So it seems like it would be fair to conduct enhanced surveillance and different kinds of abatement measures for several months after a detection.
Okay, thank you.
Yep.
Plant quarantine, given the intended use that the applicant has for in this particular circumstances and incipient population, do you feel that there is a need to make modifications to permit condition 1?
I don't personally believe that there's a need to amend that permit condition, but if the committee thinks that it's a good idea, we certainly can do that. But a population doesn't have to mean like, you know, it's established over the whole island. It could very well just mean an incipient population, or even if you have a detection, you can kind of.
I guess what I want to point out is a detection doesn't necessarily mean even an incipient population.
Again, that's up to the committee. We can definitely make changes. If that's what you guys want, now would be the time to suggest making those changes. We can certainly add detections in there.
I'll pose the question to the applicant. Does Department of Health want to see any revisions?
Well, I actually have a rather technical question. These permits that are issued, what is the lifespan of this permit?
They're generally for a year. You can either get a single use permit, which is good for one shipment for you, or you could get multiple use permits. Multiple use permits cost more, but basically if you're gonna make 5 shipments or more, it's worth it.
I guess I would advocate for having greater flexibility. I mean, you know, as Chris Jacobson mentioned, if we're talking about months, and if we're looking at detection levels versus an established level, you know, I'm not, Chris Jacobson, maybe you can help me on this. I'm not sure how long the extent of our response would be.
Well, here's a hypothetical to help me with this issue. If in 1 month you see in your port monitoring programs around Honolulu International Airport, you see trap detections of Aedes aegypti, and you know, in your weekly monitorings, you see it slowly tapering down. Is the intent to release? At what point does Department of Health kick in and say you want to conduct releases of Wolbachia incompatible mosquitoes into the environment in order to ensure there is no establishment? So, and if there's no answer for that, that's fine. Then I think we need to make adjustments in the language to create the flexibility, because I don't necessarily want to have you come back to the Board of Agriculture and go through this process in order to modify permit conditions to reflect the need that you haven't accounted for, especially since this proposal would entail only releasing male mosquitoes.
I agree.
Sorry, Darcy, can I just ask?
Yes.
Are you guys talking about the sentence, transport to or release on any island that does not have a population of the respective species is prohibited?
Yes.
Okay, so this looks like a very general statement without any parameters on how the discretion is going to be to make that determination. So I would think that only makes sense that you're bringing it up as a concern if the board is going to make set parameters to allow, so then you kind of remove the discretion by saying what this means. Because right now it's allowing discretion to both the board, I think, and maybe even to the Department of Health to kind of work out. I mean, to me, it just means that I think they don't want them to release the species where there's no population there, because they don't want to introduce it to a place where it's not. So it doesn't say how that's established. And right now there's discretion. And if you guys want to limit the discretion and decide, you're going to decide it now, like we're going to decide what it is and not allow that to be, then that makes sense. But if you want them to have it, I think I wouldn't change this. But that's all, that's the way that I read this, is that it isn't set in stone how that's established. Does that make sense?
Perfectly, at least to me.
But again, the committee can make a recommendation to have it be set a certain way.
Any other, given that, I'm comfortable leaving the language as is. Are there any other questions or concerns? Then would anyone like to propose a motion? If not, I will move to.
Darcy, excuse me. Are
Yes,
we
Grace.
making the similar changes regarding progeny in the?
Yes, that was my intent.
Okay, thank you.
Yeah, so, oh, Dr. Haas, was there many misspellings in the title this time?
I think there were actually. I have to go back and look.
Okay. Okay.
I'm sure it's just a cutting and pasting error.
Yeah. So I move that we recommend approval for the request to preliminary review the currently unlisted yellow fever mosquito, Aedes aegypti, the 3 mosquitoes by the State Department of Health, with changes to the misspellings in the title, permit conditions 6, 7, and I carefully laid out. Oh, I'm sorry. 7, 8, and 16, removing progeny. Changes to permit condition 6, which would clarify that about the trained and certified personnel being reported on. My God, that sounds horrible the way I wrote it. That there would be a report of the individuals that are trained and certified. Be approved. I am not Ken Redmond or Rob Hoff and being good at motions. Do I have a second?
I'll second.
Okay, any further discussion? So I'll take the vote. Chair votes aye. Mr. Eisen?
Aye.
Cynthia?
Aye.
Dr. Gahn?
Aye.
Mr. Fisher?
Aye.
Ms. Simmons?
Aye.
Dr. Hawes?
Aye.
Okay, thank you. Motion carries. So that's an end to that business. Thank you, Mr. Kishimoto, for your expertise in presenting these. We are now on agenda item 5, adjournment. Do I have any concerns or questions about adjourning? There being none, any motion to adjourn?
So moved.
Seconded.
Cynthia seconds. All in favor, say aye.
Aye.
The vote is unanimous, and so this. Any problems, Jennifer?
No.
Okay. This meeting is adjourned at 3:24 p.m. Thank you.
All right. Stop recording.
Thanks everyone for their time.
Thank you.
Yeah. Mahalo, everybody.
I'll stick around if anyone